Thank you for the introduction. Yeah, welcome to the talk Genomics on FHIR. My name is Patrick. I will speak slowly as people are still joining. And I see a lot of first-time attendees ribbons, which is very nice to see these. The first part of this presentation, if you were here, I think it was the same room 2 years ago at the DevDays. The first part of the presentation is quite similar because it's an introduction to the Genomics IG, and some things don't change in 2 years, but others changed significantly. So this will be the second part, the updates of the clinical Genomics IG, STU3, R4, R5, R6, resources of the genomics workgroups, and so on. So who am I? My name is Patrick Werner. I'm a trainer, consultant, implementer for FHIR. I'm currently employed at FHIR GmbH in Germany, helping companies to implement their FHIR solutions. I was with the Moelis Institute in Heilbronn before, where we designed and implemented a virtual tumor board software solution. So this is where my genomics experience is coming from. I'm doing FHIR for quite a while already. I started doing FHIR during the STU2 ballot phase, so it should be around 2019, if I remember correctly. And yeah, this got me on FHIR. I learned about FHIR at the university where I was studying. Simone Heckmann was one of our tutors, and yeah, I started with FHIR and never stopped, basically. And now I'm doing FHIR every day, almost 24/7. I'm one of the co-chairs of the Genomics workgroup at HL7 International. This workgroup is responsible for the implementation guide, for our genomic reporting implementation guide you might have heard of already, and for some genomic-related resources as well. In Germany, in HL7 Germany, I'm one of the terminology co-chairs. Elisabeth is also in the room. She's the chair of this group. And in my free time, I'm also a happy FHIR code contributor or FHIR core contributor. So basically, if you're a FHIR implementer, you know this. You implement something, you find a bug, you try to fix the bug, and this makes you a contributor if you do a pull request, which will be merged. So what would we have a look at today? Learning objectives. I want to talk about the motivation and the significance of genomics in FHIR, about the structure of the implementation guide, the overall structures, and also about the profiles which are part of our implementation guide. I brought with me a reporting example which touches not everything about this implementation guide, but covers some resources or some profiles. So, as a disclaimer, genomics is complex. The genomic implementation guide is also complex. We won't be able to cover everything in 45 minutes or 35 minutes. Then I will talk about the STU3 updates, and this GRIG stands for Genomic Reporting Implementation Guide. If I mention GRIG, it's the implementation guide. And I will talk about the updates for the resources in R5 and R6. So why are we doing this? Why should we do genomics on FHIR? From time to time, if I talk with bioinformaticists, they say, yeah, we already have standards for genomics. Why do we need FHIR? The main motivation at the beginning was modernized genomic lab reports. That's why the first product of the genomic workgroup or genomic reporting workgroup is the lab report implementation guide about how do I report a lab report in a structured manner. If you have a structured report, you can have big data, machine learning approaches, clinical decision making, which is not possible with PDF-based reports. And you also can then have some downstream analysis or research or even reanalysis years later if you have all this data. Sure, you can do the same analysis on FASTA or VCF files as well, but there you don't have the context of the encounter of the patient, other disease or comorbidities of this patient. Using genomics, especially oftentimes in oncology, is called precision medicine. So we want to be precise in the treatment of the patient. We see the disease, the tumor, as its own entity. So before precision medicine, personalized medicine, you had only like organ-focused treatments. This is a lung tumor, this is a liver cancer. With personalized medicine, you do a genomic analysis of the tumor and then decide upon the genomic profile of this tumor on the correct treatment, independent oftentimes from the location of this tumor. For these analyses, these analyses will be discussed in something called a tumor board, in this case a molecular tumor board. And preparing molecular tumor boards, so this was one of the motivations at the Molit Institute, introduces a huge workload on all the involved physicians and assistants. So when we began the work at the Molit Institute, there was some estimation that for a single case for the molecular tumor board, the preparation time for one patient is something between 1 hour up to 8 hours if it's a really complex case, because you have a lot of research, lookups in papers, the correct treatment. So having a lot of data also introduced a lot of work, and this work has to be supported or should be supported by some kind of software. The current state, and I presented this same slide 2 years ago, the current state, most of the time, it's still true, unfortunately, is PDFs. The majority of all genomic reports are PDFs, sometimes even put in fax machines, which is slowly dying in Germany due to data privacy regulation, which is nice, but still PDFs. Often they are printed out, then they are signed by a physician, then they are scanned back in, then they are in the hospital information system, then the chief physician has to sign it as well, then there will be another printout, another scanning, and we encountered some documents which were printed and scanned back into the system 3 or 4 times because somebody had to do a signature on it. This reduces the quality of the PDF significantly, and even if it was OCR at the beginning, due to all the scanning and printing and scanning, you lose all this information. All these PDFs are not standardized between labs. So if a tumor board has a new lab, the structure is differently or differs from the other lab most of the time. And this requires expert knowledge to read. I'm not saying doing genomics on FHIR doesn't require expert knowledge anymore. That would be a lie. But you can introduce some toolings which will help to manage all of this information and to the right lookups. And a lot of this tooling is still developed at the Molit Institute. So I see 2 former colleagues of mine. If you're interested in toolings around genomics, talk to them. But the solution or what you have to do before you can do nice machine-aided decision support, you have to have structured data. So that's why the genomic working group started to have a look what's out there on the PDF, which information is on PDFs, and this graph is brought from the eMERGE project, which was a very nice project. They took existing, so this is a Baylor report, they took existing reports in the PDF form and started to classify and annotate all the information bits which are in it. And then we started to map these to our existing resources and profiles. And through this mapping exercise, I would call it, we identified some gaps in our profiles, which led to improvement, the SGO2 of the implementation guide, and now to the 3rd release of the Clinical Genomics Reporting Implementation guide. To get a better idea on what's part of such a report, I brought you some examples here. So this is an example oncology report. I was working in oncology, so therefore I decided to use this oncology report. This consists of a diagnostic report, which is the broad structure, so pointing to all the relevant observations, pointing to the patient, to the involved practitioners, which specimens were used, which variants were identified, or sometimes called mutations, so which parts of the genome was changed compared to a reference genome. Then we have other observations which are more like tumor markers like TMB, MSI, and I also have a variant, a specific variant, in one of the upcoming slides. And here you can see component. This is observation.component. If you study the Genomic Reporting Implementation Guide, you will quickly find out that we love components because it's very useful for genomics. We can have one observation which has a code which represents what you're looking at, and then the components we're using it like a key value store, basically. So we have one component for, but we'll get to this. So for example, here we have the gene studied ID component. This tells you which gene was studied, and this was this ARID 1A. And then we have some DNA change in each HGVS coded, so this is a value codable concept, and the coding inside of it will be at position 6447, there is a deletion of C. And this is how we represent most of this. A lot of in the variant profile, but we also have other profiles. But first, back to the workgroup. I put some links on this slide because, yeah, as I said, it's too much to cover in one talk. If you're interested, have a look at our workgroup home at the HL7 Confluence. I also put a link to our stream in Zulip, to the genomics stream. We also have a mailing list, ClinGenomics, at lists.hl7.org. As products, we don't only have the implementation guide. We have, before we started with the implementation guide, there was the domain analysis first. So in HL7 International, you start most of the time with the domain analysis model to investigate into the domain, what do we want to model and achieve. And then we created the implementation guide, and the current version is SU3, which was finalized in December last year. And here I also put a link to this implementation guide. So this is hl7.org/FHIR/UV, which stands for universal. So it's not US core scoped, it's international. This has some implications as we can't use SNOMED-CT because SNOMED-CT is a licensed, you need a license to use SNOMED-CT, and in international implementation guides, most of the time you try to not use like licensed terminology, which are not available in every country for free. So this Genomics Reporting Implementation Guide covers all aspects of human genetic reporting. For now, it's only human. Of course, you could use it for animals as well, but now we have requests for genomic representations of viruses, for example. This is new to the group. We started focused on subject is a patient, and this is relevant for the structure, also for the implementation guide, but we are also investigating on how to represent other genomic artifacts which are not coming from a human genetic. We try to model everything that you can represent variations to the genome of a human being. For different use cases, a lot of the use cases are oncology, because oncology was one of the first areas where genomic analysis were done in a huge way, or many patients got some genomic analysis, but there are others, like pharmacogenomics is also very important if patients are responding to a substance or not, or even for transplants, it's very important, HLA, so the how's it called, bone marrow donation. So if you get a bone marrow transplant, then your tissue marker have to be compatible, your HLA markers, and this is also done with these profiles which we are having in our implementation guide. You can do full partial DNA sequencing, so all the profiles are suitable for both. You also could do whole genome or exome studies with these profiles. For now, we are focused on the genomic test results representation, but we're now also investigating for metadata like genomic study, which we will touch later. Yeah, I already told you it's international in scope. We are using free available terminologies, which is not always easy. We try to maximize the usage of observation diagnostic report to not introduce too many new resources. We introduce new resources, but we are also using observation. Main motivation was to not reinvent the wheel and have another resource in the FHIR spec. But also one other motivation was feedback from vendors, which were saying, oh, this new molecular sequence resource is nice, but it's not supported in our system. And observations are supported everywhere. So that's why we decided to use easy resources, profiles on easy resources which are implemented broadly. We can have different information amounts in these reports. As I said, we can have whole exome sequencing. We can have just some specialized genes. So some years ago in Germany, if you have got cancer, you only got the right to get analysis of, I think, 10 genes or something. So these panels are now growing. But yeah, the Genomics Reporting Implementation Guide supports all of this and can be used there. We use observations. We use a lot of observation. We try to separate them into useful parts to maximize discoverability and queryability of the data. So it's not just one big resource. We try to keep it separate, one for the variant, for example, one for the implications. We also try to be aligned with the version 2 genetic variation model to maximize consistency. So this was also one of the outcomes of the group. Before I joined, they had the version 2 reporting implementation guide, and we reused most of the LOINC mappings or LOINC codes there, so you can have an easy transition from version 2 to FHIR. So, if I want to do a tumor board, I have this workflow. I first have to have an order. All the order entry management is not yet covered by the group. There are other IGs covering order entry workflows, which should or could be combined with the Genomic Implementation Guide. This request is sent to the Genomic Lab, and this was from how we did it at the Molit Institute. This was a manual request. This was not FHIR-based, just the ordinary paper-based request to the lab together with the specimen. The lab then returned the genomic data, at first in a proprietary CVS format, but then we helped the lab to shift from the VCF format to a FHIR-based format, and then they just sent the FHIR data to us. Then this data is persisted for the patient, and on the next tumor board appointment, this patient and other patients will be discussed. This is a screenshot of the V2 of the software from the Molit Institute, where here you can see several physicians or participants of this tumor board. And yeah, right now they're looking not on FHIR resource, obviously they're looking at some imaging parts, but this conference tooling also helped to have this molecular tumor boards, which are broader or covering more than a single hospital. So this was part of a hospital network, and then it's very nice to have a virtual environment so people don't have to travel. Here you can see finally some FHIR, some representation or rendering of FHIR. This is also done by a component developer at the Molit Institute. Here you can see the different genes with the type of the variant, the source class, and so on, the transcript, which positions were changed, and so on. And also some links to CBioPortal, for example, to have a look up to get more information. The Genomics Reporting Implementation Guide, I already told you, was released in STU3 at the 12th December of last year. I'll put the link here as well. And we are covering several topics. So now we are diving into the profile and how all the data is structured and combined together to form this report. So we have this the genomics report, which is a profile on the diagnostic report. And then through the result array, we are referencing several profiles or instances, for example, the genomic findings, most importantly, variants, but we also have profiles for haplotypes or genotypes. Then we have the implication, diagnostic, therapeutic implication, molecular consequences, and genomic annotations. Then there's the rather new molecular biomarkers. This was recently introduced, and other genomic results like the sequence phase relations. But you can also, this is still a diagnostic report, you can also link other observations like some blood analysis or imaging studies or something similar. We have some extensions to be able to link to the genomic study. The genomic study, I will talk about this later as well, is a profiled procedure. Genomic study is a resource introduced in R5. Our report IG is still on R4 because most of the implementers are still using R4, and therefore we created a backport as a profile on procedure. And we have an extension for recommendations, recommended actions. This could be a follow-up recommendation or some medication recommendations. This recommendation space is still, it's usable, but if you really dig into like oncology recommendations, there you have first line, second line, third line. If this doesn't work, after 2 weeks, change to, so this needs some more elaborated like plan definitions or care plans. This is just for easy use cases. I've seen this, patient should take medication XY. Then we have a more broader overview about the relationship between the resources. We've just seen this, the genomic report, which is in the center, linking to all the different other resources. Of course, the subject, the patient. We can have risk assessments. So is the patient in risk of a certain disease, for example? The report can be based on a service request in order to represent the order entry workflow. The service request has an organization, which is a different organization than the performer usually of the genomics report. We have some supporting information. We have the specimen information. And here we have all the genomic results through result, like in the previous slide, the variants, the implications, and so on. And yeah, the recommendations and the generic study were also on the previous slide as well. Now we're talking about, or I want to talk about findings. So this is still diagnostic report dot result pointing to several different profiles. We have an abstract profile which is called genomic finding, and this is for all findings, as the name says. This can be a variant, so some mutation on the gene, but also the haplotype or genotype, which puts together several variants and then several haplotypes, or sequence phase relationships. This genomic finding is also inherited from the genomic base observation abstract profile, so we try to have like a nice architecture to reuse defined fields in downstream resources as well. Then we have the implications, the diagnostic implication, the therapeutic implication. We have the molecular consequence and the genomic annotation. I will talk about these in the upcoming slides as well. Here you can see another representation of the findings, still derived from genomic base, genomic finding. And here you can see the relation I just mentioned. We have a variant, and the haplotype is deriving from 0 is wrong, 1 to many variants. And then the genotype itself is also derived from the haplotype or can be derived from the variant directly. And we have the sequence phase relationship in between, which then can point to haplotypes and variants as well to get you an idea about the sequence phase relationship. And the last slide with a lot of boxes. Now we're looking at the implication side. So we read the findings, now we're at the implication. Still the same base profile, genomic base observation, genomic implication, and there we have the therapeutic, diagnostic, molecular, and generic annotation, all deriving from the genomic implication. And these genomic implications can generally, so all these as well, can be derived from findings, but also from biomarkers, or a combination from biomarkers and findings, or also a combination of non-genomic observations, biomarkers, findings. So yeah, really free here. So this is the abstract profile. I'm a little bit slow, so I will skip this slide. Talking about codable concepts, codings, and text. So most of the time, or not most of the time, but yeah, we support text-only representations of coded formats. I don't like it, but that's the reality. We always try to say, please provide a coded representation of the data, because CDS and all of this machine learning really likes to have structured data, but there are still use cases where labs saying, I can't do that. And the fallback for this is value codable concept.text, where you can just put into the text, but we're always encouraging people to use the correct encoding, not only to use text, but it's technically possible. Here we can see, I told you we love components. So here, some components we are using. Coding HGVS, so some HGVS representation of the mutation. We have the reference sequence, the transcript reference sequence. We have exact start end, inner start end, outer start end for structural variations where you don't really know where does it start. So we can also support a range of it. Which coordinate system are you using? Ref and alt allele for VCF style reporting, sample allelic frequency, read depth, state, and so on. So you don't have to use all of them, but you can. And that's also a challenge for lab if they are starting the FHIR reporting of genomic reports. Which components do I need? So this is the first step. You have to map your information as I have shown you at one of the first slides. This PDF part goes into this profile, into this component. Implications, somatic implications. So we have the diagnostic implication. Simple example, do the findings indicate cancer? This would be a diagnostic implication. In contrast, we have the therapeutic implication. Do they imply positive or negative outcomes for medication or therapies? So is this tumor susceptible for this treatment? Or how is the medication likely to affect the tumor based on the findings? Now I want to present you a short example of an oncology report. We started with it before, patient, practitioner, and specimen. It's just a big transaction bundle in this example. We started with transaction bundles. Be careful with transaction bundles. Transaction bundles can contain delete or updates and mess your data. So be sure to filter them before you apply them. So we have a huge bundle. We have the practitioner which did the analysis. Then we have the specimen about most of the time the tumor, or sometimes the blood sample from the patient. We have several observations. So this is a tumor marker, the TMB, in a line-coded way. So most of the time we are using line if possible. If it's not possible, we like to use NCIT, sometimes others, but most of the time line if available. And here you can see the TMB is low, as an example. Then we have the MSI, the microsatellite instability, also line-coded MSI here. Subject still the same patient, and MSI is stable in this patient. Then we have the variant of ARIDA 1A. We have a DNA change here, and also this is coded DNA change with the appropriate line code. And yeah, as I mentioned before, a deletion at a certain position. So all this together then, so here these are other results, just an overview. You have the diagnostic report pointing to all of these observations most of the time. This altogether forms the genomic report. Heading over to the updates to the genomic report IG. The SU3 introduced new operations, so we have some operations defined in our IG. We added some new ones and some better guidance and extensive examples. We added the support for genomic study, which is similar to imaging study, more or less. It is able to capture structured study-level metadata, so what have I done in the lab, basically. We broadened the support for biomarkers. Biomarker observations were not part of the IG. We introduced them with SU3 with a very generic molecular biomarker profile. We updated the representation of molecular consequences through a new molecular consequences profile. Before it was part of the variant, but now we are separated the consequences from the variant because semantically it just makes sense. One is this changed, and the implications is what do we infer from it. And we improved the guidance on the somatic reporting page. So we have a detailed synthetic report, additional example resources in order to easier understand all of the possibilities. I mentioned before we have the backport, the genomic study, which is a backport of an R5 resource, a profiled procedure, which by itself references here a study analysis, and this study analysis is also a profile procedure. As you can see, a lot of extensions. This is just used to be able to backport all the information. Here you can see which method was used, with which genome build, which focus specimen, and so on, to just have a representation what was done in the lab. We have the biomarker profile, which is a small profile. Category is lab. This is biomarker, and this is genomics. We have a code, or several codes with an example binding. You can use your learn codes for MSI or whatever you prefer here. And then we define some components like which gene was studied or what kind of biomarker are we looking at. So these biomarker categories is a code system. Could be antibody, antigen, lipid biomarker, protein-based biomarker. So this is just some classification for the several different biomarkers. Then we have the molecular consequences profile, which took out some of the parts of the variation profile to be represented in its own profile. HGVS, transcripts, protein HGVS. So this is also used a lot of times with calculated results. So you just see the genomic variation and you don't necessarily see the protein changes, you just calculate it. And this is then put into the molecular consequence profile, which is derived also from the genomic implication. Short look at improved somatic guidance, a little bit like eMERGE, not as colorful, but with numbers. There we tried to give an idea. This is a real report, a synthetic, but a real, based on real data report. And we put in all the numbers so you can have a quick look what goes in which box, basically. The genomic annotation is also another new profile. It's not in SU3, it's rather new. This is mostly used for population allele frequency at the moment. We plan to use it for miscellaneous genomic annotation, so everything which is not a diagnostic, therapeutic, or molecular consequence will then be a genomic annotation. Quick look here, genomic implication, observation profile. We love observation, I mentioned that before. And here we can have, for example, down the population allele frequency is down here. And there's also an ancestry group, so all this population-based information goes into this profile. We also had updates in the genomic FHIR core resources. Molecular sequence will be removed or merged with molecular definition. we have genomic study, I already told you that, and we have molecular definition as a new resource. Genomic study, I will skip. Molecular definition is a new resource meant to represent multiple different things, like DNA, RNA, proteins, in a definitional way. Because the observation is just the observation, it's not a definitional resource, it's not an observation definition. And to capture this, we will introduce, or we already introduced, molecular definition. It's an abstract resource which needs profiles in order to be useful. So we are currently creating an implementation guide just explaining how to use this resource. Genomics is complex, and therefore we have a resource with its own implementation guide in order to be able to understand what's possible with it. Quick overlook here. So this is the future idea. We still have like observations, observed molecular definitions, which are then pointing out to these profiled molecular definition resource like allele, sequence, or variation. These will then be profiles on molecular definition. And you can see several here. It starts with sequence or allele, so you can use it to just represent your sequence, AC, AAC. You can have a contextualized sequence, which is then called allele. Then we have the variations and the adjacency of these variations. Then we can say these variants form this haplotype or gene fusion representation. Features, biomarkers, genotypes, all of these will be profiles on this molecular definition resource. So this is what we're currently doing, working on implications and annotations, updating on the core resources. We have to have a look at family member history soon as well. It's a version 3 IG which was retired, but I personally think we still need this in FHIR. Population allele frequencies is one of the hottest, newest topics, and we need to build this IG around molecular definition to be able to show you how to use it, basically. So, sorry, I had to speed up a little bit. I was too slow at the beginning. What did we learn today? I showed you the FHIR genomics motivation significance, how to use the reporting IG, have a short look at examples, current topics of IRG, updates in STU3, GRIG, and R5, R6 FHIR genomic resources. I'd like you to get involved, join our chat, join the web calls. We are always seeking for input. You can contact me by mail via Sulip. I want to thank you all to all contributors of the workgroup, also to all my fellow co-chairs, which provided a lot of input to this presentation as well. So thank you.
Thanks a lot, Patrick. That was a very informative session. We have time for some questions. There are no questions on the FHIR, but anyone would like to go first?
Thanks for your talk. Just quickly imagine, like, if we have a VCF file with 1,000 mutation in it, how many FHIR resources are needed to represent that VCF file with 1,000 mutations?
It depends. If you have 1,000 variants, it would be 1,000 variation profile, 1,000 observation resources plus implications, so a lot. This can be a problem. I spoke very briefly about the new operations. The idea behind the operations is that if you have huge data repositories, you maybe have already an established data format in your repository, and these operations are then used to facade your existing solution to be able to query it. So your variants still might be in other formats like VCF, but these FHIR operations will enable real-time translation of these into the FHIR resources. But yes, a lot of variants means a lot of resources.
So basically a whole genome VCF file will be millions of resources.
Yes, yes. So this is more suited for reporting. That's why our implementation guide is called reporting. Report the significant or the important parts of the... I know there are a lot of variants with unknown significance, and most of the times in a hospital environment, nobody cares about them. And that's why at some point you have to decide where you enter the FHIR world. But for whole genome sequencing, it would be not beneficial to just put everything into FHIR. Yes.
Thank you, Patrick. Yes.
Can you just briefly compare molecular definition to observation definition?
So the molecular definition is a container resource, basically. As you can see, you can profile it to do everything, mostly. So the observation definition is, I'm not an expert in observation definition, but if I remember it correctly, it's about an abstract definition about a way of how to examine several values of a patient, right? The molecular definition is a definition of resources around genomics. So we have some fields where you could put in sequences directly in string representation, but also sequences to link it out to another repository, which touches the first question. So you can have a molecular definition profile having just pointers to binary formats for the actual sequencing data, but have a structured way of representing only the variants or the variations to these, which are included in these binary files, basically. So not a real good answer to your question, I'm sorry, but I'm not an expert in observation definition.
Anyone else for questions?
Thank you for the talk. So the lab need to write the data report along with this IG. So I would like to know the current status of social implementation with lab. Social implementation.
Yeah.
So lab needs to write the report along with this IG.
Yeah.
So it's a key point to adapt this IG.
Yes. We try to be aligned with GE4GH and other initiatives so that we have like the similar ideas or parts. But yeah, you have to model it to be compatible with the IG. That's what we done at the institute before. As I said, at first we created some translation software from their proprietary format in our format. But during the implementation of this translation layer, the lab got interested in FHIR and saw the benefit, and we provided them with some code snippets. And yeah, if you have like templates or examples, it's not that complicated, more or less. Yeah.
We have room for one more question. Anyone?
Hi, I have a question about the implications which are stored in the records. How do these relate to the CDS hooks, which are also possible in FHIR? Like, should CDS create these resources?
Could be, yes. So when we implemented it, most of the CDS part was just showing information to the physician to give him a better overview of the actual state of the art, basically. But yeah, you can elaborate these CDS hooks, and one possibility would be have a look at the variants, all the observations, derive some implications from it, and in your CDS hook, implement them as implication profiles and feed them back to the system. Yeah, that would be possible in the decide workflow.
Thank you.
Welcome.